So I'm going to talk about genetics today, and I have a picture here that I took from the deck of my little mountain place of my gardens that I got going. I'm really proud of my little gardens there. And one of the things that I've been doing that I think is really interesting is I've got several tomato plants, and there's some different varieties. And one of the fun things to do is cross-pollinate some of these tomato plants and get different hybrids of tomato to see which ones are the best and which are most flavorful, which ones get the biggest. And this idea of making hybrids and manipulating genetics has been around for a long time. How does that happen? How can we get hybrids, and how can we take different plant species and mix them together and get some different version? In this video, we're going to take a look at genetics, and the first person who did that, Mendel. Let's get started. So I mentioned that a lot of this started with who we consider to be the founder of genetics, Gregor Mendel. He was a monk that lived 1822 to 1884, and he really started this whole discussion with work he did with pea plants. So Mendel took some pea plants, right, some plants that make peas, and he cross-pollinated some true breeding parents. True breeding just means, we'll find out later, that they have both of the same gene, both of the same allele. And he cross-breeded or cross-pollinated some of these parents, raised the seeds, observed the traits in the next generation, which we would call the F1 generation, and then allowed those to pollinate, and then we got the F2. So what he did was he noticed, oh, okay, we've got some pea plants, some of them have white flowers, some of them have purple flowers, and he wanted to do some work to see, what if you crossed this, take some of the pollen and put it over here on this, take some of the pollen from that and cross it with another. What do you get when you cross-pollinate or sort of make a hybrid of these plants? And he found some really interesting results. So what did he find? What he found was that when he took a true breeding purple flower pea plant, right, you got the purple flower, crossed it with a true breeding white flower pea plant, see the white flower, he thought, okay, maybe I'll get a little mixture of both. But from the parent generation cross to the first generation, or the filial one generation, he found that they were 100% purple flower. What's going on there? What happened to the white flowers? There were none in this F1 generation. Well, those actually happen to be hybrids, right? Like the tomato plants I were talking about earlier. And what we found was that 100% of those were purple. Okay, so he did some more. He took that generation, 100% purple flower pea plants, let them self-pollinate, right? So took the pollen from one of these, and right, so they were self-pollinating only with this generation. When that was allowed to happen, then we finally saw the white flower pea plant again, but it was only in about 25% of the resulting. So in other words, we had a 3-to-1 ratio of purple to white flowers in the F2, the filial 2 generation. What's going on? What was happening there? Well, let's look a little deeper. What did these findings mean? Well, what we now know is that traits come in alternative versions. So purple versus white flower color, right? We're still talking about trait flower color, but there's two versions, the purple version and the white version. The fancy genetics term for that is alleles. An allele is a different version of a gene on homologous chromosomes. So if you have two chromosomes, like if chromosomes, if it's a diploid organism, and you've got a gene for flower color, one chromosome might have what we call a dominant, and one might have what we call a recessive gene. These are different alleles for this trait flower color. Now what's different? Well, if you look at the dominant versus the recessive, there's actually a different nucleotide sequence when you look at that gene. So different bases, right? Adenine, thymine, cytosine, guanine. You might find a different one at these different nucleotides. So, and here I actually have a nice picture of what I just drew up there. you can see on a homologous pair of chromosomes, right? So let's say these are both number 7 chromosomes, right? In humans, we have 23 pair. Let's just say we're talking about number 7. In flowers, in the pea plant, I'm not sure exactly how many chromosomes, but we'll just say it's number 7. One of these has the allele for purple flowers. One of these has the allele for white flowers. It's the same pair of chromosomes, right? They're both number 7 or number 2 or whatever. But one of them has a slightly different version. This one has a purple flower allele. This one has a white flower allele. So, why then did purple overshadow the white? Well, this sets up what we call dominant versus recessive alleles because some traits mask others. And in the case of Mendel, the purple masked the white. It's sort of like, I like to think of this as like if you know a couple or two people that are together and maybe one's a little more dominant, one's always doing the talking, and one is there, they could express themselves, but they maybe don't. That's kind of like what's happening here, right? So if one chromosome is saying, quiet, I'll speak for both of us, right? The white allele's still there, but the purple one's speaking for it. That's when we have a completely dominant situation like we have here. So the dominant allele is fully expressed, and when we're writing that out, we'll use a capital letter. In this case, we're going to use a capital letter P for a purple pea plant. So if one's dominant, the other is recessive. So the white one, in this case, must have been recessive. We didn't see any noticeable effect from the white allele. And in some cases, in many cases, the gene, the recessive gene, makes a non-functional protein for, in this case, color, so you didn't even notice the effect. For recessive alleles, we'll use a lower case letter to indicate recessive. Okay? So dominant versus recessive. Dominant, if it's present, is expressed. Recessive is not expressed if the dominant is present. Now, if something were to happen, if the dominant was damaged or not there, then the white allele would have to say, okay, I'll speak up now because the other one was damaged and wasn't expressing itself. And when you do these crosses, right, in this case, the purple versus the white, what we saw was that purple must have been dominant because it's the only one that's expressed in that first generation. So now I want to talk about two vocabulary terms, genotype versus phenotype. And what we find here is that phenotype is the physical description of an organism's trait, whereas the genotype is just the description of the genes or the genetic makeup. So in the case of these flowers, right, let's think about the genotype and the phenotype. We'll start with phenotype. What is the phenotype of that flower? Okay, so let's think. The phenotype, it's purple, so the phenotype would be purple. Yeah, purple. That should be an exclamation. Purple. Over here, what's the phenotype? Oh, white flower. Now the genotype is a little more difficult. That's going to be the genes. So in this case, because purple, when crossed with white, made all purple, we're going to say that these must have some dominant alleles. These must have some recessive alleles. Okay? So we'll get into the genotype and give you the specifics of how we say this here in a moment. Okay, so let's look a little deeper at genotypes. Two vocabulary words I want you to learn is homozygous and heterozygous. When you say homo, you think same. So homozygous means the same alleles. Either they're both dominant or both recessive, both uppercase P's or both lowercase P's, in this case of the pea plants. For heterozygous, they're different alleles. So you got one dominant and one recessive. Now, let's look and try to put the genotypes for these flowers. So here's the purple. We know it's dominant. It's got at least one big P. So our two options are either homozygous dominant or heterozygous, right? Because we know as long as there's one big P, one of the dominant alleles, that will be expressed. Well, what about the white one over here? Well, we know that because this is recessive, has to be both lowercase p's or both homozygous recessive genes. Okay? Okay, now, when we want to do a cross, when we want to cross two of these, in this case flowers, we'll do what's called a Punnett square. You've probably seen these before. But let's say we wanted to cross both of these flowers. Both of them are heterozygous, right? One big P, one little p, crossed with one big P, one little p. So let's do the cross. When we do this, we set up, if it's just one trait we're looking at, which is looking at flower color, we set up sort of a 2 by 2 box, 4 squares total. And what we do is we take the male and the female and put them on each side. So let's just say for kicks, this one over here is going to be the pollen or the male sperm. So we'll take that and we'll put it right here. We take one of them in there. We take the other one, put it over there. If this was the female, the egg, we're going to put that there, one there, and there. Then you just do the cross. So this first one would be a big P and a big P. And then we would know that that's, of course, a purple flower because it's got the dominant. Over here, we're going to see one big P and one little p, also purple because we have at least one dominant purple gene. Down here, we got one big P, one little p, also purple. But over here for this final one, when we take this P and this P, we get, right, 2 little p's or homozygous recessive. That means this is recessive or white. Now, if we were then to take and look at the genotypic ratio and the phenotypic ratio, we could just line them up, right? So that's this. I've just moved it over there and lined them up. We see that there's sort of a 3 to 1 phenotypic ratio, 3 purple to 1 white. There is a, as you can see, 1 to 2 to 1 genotypic ratio. And we're looking at genotype, we're looking at these, right? So there's 1 homozygous dominant, there's 2 heterozygous, 50%, and 1 homozygous recessive. So for genotype, it's 1 to 2 to 1. But when we're looking at phenotype, the physical characteristic, we're looking at 1, 2, 3, and 1, 3 purple to 1 white. So you need to be able to do a Punnett square and then think about what is the genotypic and what is the phenotypic ratio that we get as a result from the cross. So now we come to a problem. Let's say that you're given a purple flower and someone says, okay, we know purple's dominant, that's the dominant phenotype, but what's the genotype? Why don't you think about that for a minute? What's the genotype of that? Can you tell? Well, it's hard to tell just from looking at it, but really it's got to be one of two choices, yeah? Is it homozygous dominant or is it heterozygous? It could be either one. So how would we determine if we had a purple pea plant, how could we determine if it was homozygous dominant or heterozygous? Well, what we'd have to do is actually do what's called a test cross. So we'd physically have to take the flower and crossbreed it with a homozygous recessive, in this case that would be a white flower. So we'd take the flower in question, we'd cross it with a homozygous recessive white flower and determine the identity of the unknown allele. Because we don't know, is it this or this? So what we'd have to do is cross it there with what we do know, the genotype for, because anytime we see the white flower with these pea plants, we know it's got to be this, right? So if we did these crosses, we could figure out what the flower in question was. So how does that look? Well, to do a test cross, you got to do two. So you got to do these, and then you got to do it again and see what you get. So if we did this cross, and we took a homozygous dominant with a homozygous recessive, all right, so we put those there, then we're going to put these over there. What we'll get, of course, is 100% purple. So if we did the cross, we took our flower in question, and we crossed it with a white flower, and we got 100% purple, then we know this genotype had to be homozygous dominant. On the other hand, if it were heterozygous and we crossed it with a white flower pea plant, what would we get? Oh, we'd get some purple and some white. So we'd get sort of a 50-50, 1-to-1 ratio. So if we got this ratio, we'd know that the flower in question was actually heterozygous. So we call this a test cross because we're just doing a cross to test to see if it's homozygous dominant or heterozygous. So now let's talk a little bit about genes. We know that genes that are adjacent or close to each other on the same chromosome tend to move as a unit. So when they segregate, the ones that are close will stick together. So let me give you an example. So here we're looking at some different traits on some fruit flies. Okay, so we got some fruit flies, and we're looking at one of their chromosomes here. Okay, and if we look at the chromosome, we see that the gene here for either, it looks like kind of long antenna or short antenna, is right here at this location. Pretty close to it, oh, look at these wings. This one has like normal wings. This one kind of little dumpy wings or something, right? They don't look quite right. And then we're looking, oh, what's going on here with the legs, right? So what happens is, you may remember when crossing over occurs in meiosis. Sometimes some of the chromosomes cross over and exchange genetic information with another chromosome, and then that genetic information would cross back over. Well, if the genes are closer to each other on the chromosome, when crossing over occurs, right, when this little bit of a chromosome exchanges with this, the ones that are close together are going to go together. So these genes that you see here are more likely to stick together and be common in the results. So if you had two parent fruit flies mating and they had all these same characteristics, if the baby got this characteristic for the antenna, because these are close together, they're more than likely also going to get this characteristic and this characteristic as well. So genes that are adjacent and close to each other often move together as a unit. So in a way, they're kind of linked together, and we see them together more commonly. So we also will talk about how we get some variation. Now, these two laws are a little challenging, so I'm going to do my best to explain them as easily as possible. Two laws I want to talk about, two of these Mendelian laws. The law of independent assortment and the law of segregation. So the law of independent assortment says alleles of genes on non-homologous chromosomes will assort themselves independently when making sperm and egg. So for example, here we have two non-homologous, right? Okay, so in here, the blue, those are both homologous. That's the same. Let's say those are number 7 chromosomes, and let's say these two are both number 8s. So when we're saying non-homologous chromosomes, we're actually talking about one number 7 and one number 8. Those would be non-homologous. They're not the same. And all we're saying in this law of independent assortment is that the alleles on this one could go into this sperm, and the alleles in this one could go into this as well, or not. They assort themselves independently. So just because this one maybe had a bunch of dominants on it, right, doesn't mean the one here, maybe with a bunch of dominant genes, will also go into the same one. The alleles assort themselves independently. So that's law of independent assortment. The law of segregation says that two alleles for each gene segregate into separate sperm. So meaning, let's say this one had a dominant and this one had a recessive. You don't get both for the same, maybe we're talking about flower color. You don't get both of them in the same gene. Notice that this allele went into this one, and this one would go into here. So this one would have a lowercase, this one would have an uppercase. That's if one was a dominant, one recessive. You could have both being dominant, then they would go in. But the deal of this is that the two alleles segregate independently. So only one allele goes into each sperm or egg during meiosis. So now we're going to try to think, what if we wanted to look at more than one trait? So far we've talked about one trait, purple or white flower. But let's say we wanted to look at two traits at once and predict what the outcome will be of two parents when they cross. So we're still going to talk about pea plants, but you might know some peas, the actual pea, is round. Sometimes they're actually wrinkled, like you see there. Sometimes the pea is yellow. Sometimes the pea is green. So in this case, we're going to look at a yellow round pea, right, two traits, crossed with a green and wrinkled pea, those two traits. And let's see what the outcome will be. When you do the cross, you realize, oh, 100% of them are yellow and round. What does that tell us? Well, it tells me if they're yellow and round 100%, that probably means yellow is dominant and round is dominant, meaning yellow is dominant over green color and round shape is dominant over wrinkled shape. And so then we can assign the genes, right? So we're going to say big Y is yellow, big R will be round. So remember, you got to have two of each. That means little y will be green, little r will be wrinkled. And so this must be big Y, big Y, big R, right? Homozygous dominant for yellow. I told you they were true breeding. Homozygous dominant for round. That must mean this one, because it's recessive, has to be homozygous recessive for both traits. So when we do this cross, it makes sense that we're going to get all yellow round peas, because if you think about this, this parent here had to give a gene for color and a gene for shape. The only option they could give for this is a big Y. The only option they could give for shape was a big R. So that means every one of these has got at least one big Y and one big R. These over here, the only option they could give was a little y for color and a little r. So that means the only option in the sperm or the egg for there would have been both the recessive ones. So this is what we get in this round. Now, what if we allow those to self-pollinate? Well, we get a really interesting case. When we have a dihybrid cross from true breeding parents, and then the F1 generation is heterozygous for both traits, we get this interesting 9:3 to 3:1 ratio, where 9 of them will be yellow and round, 3 of them will be green and round, 3 will be yellow and wrinkled, 1 out of the 16 will be green and wrinkled. So this is what we get. Now I'm going to show you how the actual cross works and how we got these results. So when you're doing a dihybrid cross, you got to do a 4x4 little table here. Okay, so 16 total. The hardest part for many people is figuring out, when we're doing this cross, what are the actual alleles that each parent could give. So what you got to think is, if let's just say this is dad, and let's say that this is mom. Dad has got to give one gene for color, or one allele for color, and one for shape. Mom has also got to give one for color and one for shape. So you got to think, what are the 4 possibilities that they can give when they have to give one of each? So really what you're doing, let's take mom here at the top. So she's got to give one, so that could be a big Y with a big R. It could also be a big Y with a little r. Those are the possibilities. Mom could also give a little y with a big R, there it is, or a little y with a little r. Those are the 4 possibilities that she can give, knowing that she's got to give one for color and one for shape. Dad's got to do the same thing. He can give the big Y with the big R, the big Y with the little r, or he can give the little y with the big R or the little y with the little r, right? So these are the 4 alleles they can give in the sperm, okay? So when you do the cross, you just put them together and you keep the Y's together, keep the R's together, or whatever you're talking about. And so here's what we get, and then you can sort of just keep going. If there is one dominant, you generally always put that first. See, I would not put the smaller case before the big. Okay? So when you do that, that's what you get. Okay? You just go through, and it's just simple math, just put all the letters down correctly. Then you've got to go back and actually determine, well, is this yellow, round, wrinkled, green? What is it? Well, in this case, because we've got one dominant, at least Y, it's yellow. One dominant R, it's round. So same thing there, same thing there, same thing there, same thing there. Uh-oh. No dominant for shape. That must mean this one's wrinkled. Okay, then we got dominant again. Up, another wrinkled one, but dominant for yellow. And then you can sort of see, oh, now we're getting to here. We got 2 recessive genes for color. That must mean that one is green. Then we get a green, another green round. There we go. And then we sort of finish it out. Notice that the least common would be green and wrinkled. So when you take those and you try to make a phenotypic ratio, what you find is that we've got 9 of those, 3 of those, 3 of those, and 1 of those. So we get the 9 to 3 to 3 to 1 ratio. Now, could we do a genotypic ratio for all these? Yes, it would be very complicated and it would take up a lot of time and space. That's why for these dihybrid, we generally only ask, what's the phenotypic or the trait ratio? Okay, so hopefully this video was helpful for you, either remembering how to do some of these Punnett squares or perhaps learning for the first time. Remember, the important thing is to think this is all about alleles on chromosomes and which version of the gene each organism is getting from the parent.